of you know the central dogma of molecular. Yeah. So that's where these wonderful systems biology has really popped up in early 2000. So the word systems biology means the biology of a system. That's where you tend to use this particular word. So usually a system has got several components, the major components. Like if you take a human organism like us, you have a lot of visible parts like the eyes, hands, legs, and all those kinds of things as major components. But if you really dig deeper, human organisms has got several organs. Okay, and within those particular organs, you talk about major tissues like the liver, heart, and all those kind of things. And each tissue has got several cells. So in an organism like humans, we have got about 1 trillion cells, and each of these particular cells has got several organelles. Okay, like mitochondria, nucleus, cytoplasm, Golgi complex, okay, and all this kind of things. And there is always a kind of interaction between these organelles. So if you take about nucleus, it has got its own genome, it has got its own genetic material. If you take mitochondria, it has got its own genetic material. In case of plants, you have got some extra an organelle like chloroplast, okay, where it has got its own genome again. Okay, so each of those particular things play absolutely a kind of a vivid role. But each of those particular organelles has got those particular genes transcribed. You have got the proteins. They keep interacting with each other. And if it were some organisms like microorganisms like bacteria, there might be strains adapting and unfitting in the environment. Okay, the best thing is the SARS-CoV-2. Okay, you see, SARS-CoV-2 is the latest novel coronavirus. The SARS-CoV-1, came into existence way back in 2003. But that probably was gone within 1.5 to 2 years. But the SARS-CoV-2, it is trying to fit into the environment. Okay, and it is not yet gone. It is several strains and various entities are still evolving. Okay, so we may say that all these kind of things are nothing but the systems biology, because that's where the systems biology talks about the genes lost and found, probably the strains adapting and unfitting in the environment. So basically there are contexts in the genomes. You always talk about context. So contexts are nothing but the ohms. Okay, ohms are nothing but many. So whenever you say genome, study of many number of genes. Proteomes, study of many number of proteins. Transcriptome, study of many number of transcripts. Likewise, there are about 337 omics existing today. Okay, so if you talk about plants, you always talk about the stress traits or the quantitative trait loci or the genes that are responsible for the stress, like abiotic or biotic stress. If you talk about animals like cows or buffaloes, there might be certain things like feed efficiency traits, okay, where the animal is probably overfed, thinking that if it is overfed, probably it might give more milk, something like that. If you're talking about bacteria, we always talk about monoisolate or multi-isolate, or whether or not pathogenic or non-pathogenic. But if you talk about humans, we humans are the most intelligent people. We live across different environments. And as a result, there might be different diseased phenotypes. Maybe a person living in Canada may not really have a kind of a similar disease to that of a person living in India, and vice versa, isn't it? So the epigenetics of the environment play a very, very important role. Okay, so that is very, very important, and the common most variable across all these vivid organisms is the environment. So that will allow us to define the true positives. I'm sorry, someone says it's not clear. Is it my voice, or is it the context, or is it my video stream? Can you please?
Voice actually for some time.
Yeah. Yeah, I'm really sorry in the Priya, please bear with me. I'm really sorry. I apologize, for this. Yeah.
Please
Yeah.
continue.
So, the true positives, the true negatives and false positives and false negatives is the real context, we usually, talk about. And that's where, this wonderful discipline called systems genomics, came into picture, because we talk about the study of genes in the system. This is what we called as systems genomics, study of many number of genes. Okay, you talk not just about the genes and the context, but you also talk, their role in that particular environment, and that's how this particular systems genomics has come into picture. And there are many particular tools in systems analysis. The most important tool is this Galaxy project and biogrid.org and usegalaxy.org. There are many other tools. As you could see, all these particular tools, you can simply play around. You can zoom in and find your own gene of interest, and you can identify, the function of these particular genes, to the relative context. Okay. So a quick summary of my first part of my talk is, bioinformatics and systems genomics play a very, very important role. We need to bring, various challenges for this. We need to bring consortiums and collaborations, because this is not just done, from one particular, what you call tool, or one particular, what you call, lab, maybe one particular, what you call, organization, or one particular institute. We need, the so-called, multifaceted knowledge, multifaceted disciplinarians, coming together in the form of consortia. The best such consortia that I could really talk is about something called the FANG, Functional Annotation of Animal Genomes Consortium, which is based in Iowa State University. They were really successful in bringing. And if we identify any gene proteins in the context, the most important thing that we usually, talk here is what candidate genes and proteins. It's not just identified a gene and you have given function. Sometimes you may have to curate it very well. Okay. There are also regulatory elements, because many of these particular genes or proteins try to keep interacting with each other. That is what we call it as regulation. Okay, they don't sit single, or they don't sit idle. So if there is a gene, there is a protein, they usually keep interacting with each other. So that would probably allow us to identify the function to those particular genes and proteins. Okay, so this is how the need for so-called sequencing efforts have really come into picture. Okay, that is what we call it as the next generation sequencing, which we will probably describe. So that is how the current next generation sequencing efforts have come into picture. So the next generation sequencing is no longer next generation sequencing, because it is the now generation sequencing, because there are many, I would say, many what you call tools, many sequencing platforms that have really come these days. And the erstwhile next generation sequencing is no longer next generation sequencing. So what is next generation sequencing, by the way? This is what, probably I'll just try to describe it in the next few minutes. So what happens is each of those particular sequencing fragments are split into several DNA strands or DNA fragments like this. Okay, as you could see from the particular slide. And each of those particular strands are ligated, and adapters are added to that. So as you could see, the pink, what you call end is each of those particular single strands, a pink color, which is the particular, hybridized layer, So it's just like, since are added to each of those particular strands. Okay. And now there are also these linkers are also attached to that. Okay, and then each is 50 and it is sent to the machine, and the machine does where you can copy more number of sequencing strands, and you get those particular reads using these particular base chain reads. There are many tools like the next generation sequencing tools, depending on the type of sequencing that you do. There are different types of sequencing. Is what we call the whole exome sequencing. The other one is a whole genome sequencing. There is something genome sequencing, and there is also something called metagenome sequencing. Okay, I'll just certainly touch the base and define. Tell you, in whole genome sequencing, in a genome you got in a eukaryote in a eukaryotic system, you got both introns and exons, right? Okay, so you got exons which accumulate to about 1.8% of a human genome, for example. Okay, introns are about 98.2%. So if you want to sequence both intronic chunks or identify the mutations present in the introns and as well as exons, so you sequence the entire genome of interest. That is what we call it as a whole genome sequencing. Okay, in a whole exome sequencing, because in an exome you only have the coding chunks. Okay, because you would only sequence the coding exons. So that is what we call it as exome sequencing. So you'll only identify the mutations present in the exons. There is something called targeted genome sequencing. In a targeted genome sequencing, maybe you want to sequence some region of say chromosome 1, some regions of chromosome 2, or maybe some genes like BRCA1, maybe like TP53 only those particular genes of interest. Okay, then you can tell the service provider or the instrument saying that you only want to sequence those particular genes of interest or those regions of interest, and that's where this targeted genome sequencing comes into picture. Metagenome sequencing is nothing but meta means many. Say you try to collect some a sample say from water or sewerage, and try to identify what are the bacteria present in that. So you really don't know what are the bacteria present in that. So sequence as many number of genomes of interest. That is what we call it as metagenome sequencing. So all the next generation sequencing data, those ATGC reads that you get, are obtained in the form of FASTQ files, which we call it as fast quality files. Okay, FASTQ, FASTQ files. Okay, there is also something called, as you could see here, when you have these particular FASTQ files, at the end of the day, mutations, isn't it? So most important what we call of using the relation sequencing is from those mutations, you need to identify, screen, and jot down and identify what are those particular mutations responsible for that particular disease. And those particular files are obtained in the form of a file called VCF, which is a variant calling format file. Okay, but before which you need to annotate them, because those reads or those mutations that you get, you need to check across database. Okay, cross check and finally you got some mutations of your particular interest. Okay, so that is how you can say that these particular of it's not always that there is only open source tools, or maybe you have to do everything by yourself. Okay, there are some commercially available tools, just like Windows machine. Okay, if you use a Linux, it's an open source, but in a Windows machine you got some important what you call software like MS Word, MS Excel. Likewise, there are certain sequencing what you call tools sequencing tools very which I was just telling you. Okay. Likewise, but in bioinformatics, my dear friends, the most important thing is you need to reach consensus. You cannot say that you have used one tool, okay, and then you have gotten some results. That is not the way. Okay, you need to probably reach consensus. You need to use one or two tools, one or two vivid tools, and say that yes, you have gotten some results. Okay, that is very important. I'm not in great detail because these are all what you call the complex things and of things. What are the things? So, like when you perform an experiment, FASTQ files, but you need to quality check. You need to ask whether or not things the quality of particular sequence. Okay, these are absolutely very important. So if you know those particular, then you can probably try to say that yes, the quality is good, and then the quality is bad. And then you can probably ask the service provider, or you can ask the instrument to resequence those particular segments of interest. Okay, and if you're talking about sequencing, usually there are two ways of sequencing. You can sequence right from 5' to 3' of the strand, which we call it as a single end sequencing, which means you're sequencing the forward reads of that particular strand. Okay, but if you want to sequence both the reads, which means forward and reverse, that is what we call it as a paired end sequencing. Okay, which means it's just like you are there in a conference room. You are asked to pick up all the pencils present on the table, and you need to walk down from one end to the other end. One way you might miss certain pencils kept on the table. Okay, but if you return in the same path through that particular tables, you might pick those particular pencils. So likewise, the sequencing machine as an option for you to go for paired end sequencing, which means you sequence from one end to the other end, and again reversely you come and check and read those particular bases, just to double check. So you'll have a forward strand read and a reverse strand read. Okay, that is what we call it as a paired end sequencing, which is always a kind of a good idea. And if your lab is rich, you can also use something called a mate pair sequencing. In a mate pair sequencing, you sequence multiple reads from one end to the other end, and in the reverse also you sequence multiple times. Okay, so the most important thing, this is what I was just trying to tell you. The sequencing take home here is, you have whole genome sequencing, you've got whole transcriptome shotgun sequencing, which I forgot to tell you. In a whole transcriptome shotgun sequencing, which I believe Pushpendra will give you an idea, you take the RNA as a seed molecule of interest. Okay, you isolate RNA from your organism, you reverse transcribe to cDNA, and then you check the gene expression profiles. Okay, and then you ask how many transcripts per million reads are there, and whether or not any of these particular genes are differentially expressed. Okay, so likewise targeted genomic sequencing, the whole exome sequencing, and of course a metagenome sequencing. So this is how it is. E is exons. In between you have got only introns. So if you only sequence exons, it is whole exome sequencing. If you sequence only both exons and introns, it is whole genome sequencing. If you sequence a mixture of both introns and exons of variable interest, that is targeted genome sequencing. Okay, so I'll just tell you a very important concept in next generation sequencing. I'll just try to finish in another 10 to 15 minutes. Okay, this is a very important slide and take home message. So imagine, you know, you are paragliding, you know, from an outer space, okay, some 10 kilometers above the sea level, you are paragliding, and some 10 kilometers above the sea level, you know, you cannot see anything, right? But you want to get down somewhere in, Delhi Rajpath, okay? But you'll not see, now you are somewhere above 5 kilometers. You can see the periphery of, you know, Delhi. You can at least see, you know, some gray areas. Now you come down to about 2 kilometers above the sea level. You can absolutely see, the inner ring road and outer ring road of Delhi and all those kind of things. And now you are somewhere above 500 meters above the sea level.
Yes, so I think there is some connection issue with Dr. Prashanth's network. So just for very quickly, we wanted to cover the comparative transcriptomics, and in the meantime, if Dr. Prashanth is able to connect, we will get his views. But I'm sorry for some technical glitches. Please bear with us, and hopefully we can cover some very basic aspect of the comparative transcriptomics using next generation sequencing. So basically, let's come to some basic aspect of what is the definition of transcriptome. It's basically the entire transcript of a cell that is referred as transcriptome. In some cases, when the context is about the messenger RNA, then it could be the entire mRNA of the cell that is referred as the messenger RNA or the transcriptome. But once we have studied genome or the exome, why study transcriptome? We know that all cells behave differently, even though they have the same genome. In a human body, for example, you would have different type of cells, and they would be functioning differently because their transcripts are different at a different time point in different conditions. So accordingly, it also affects in the microorganisms, it affects the pathogenesis, it affects the drug resistance mechanisms, or gene expression profiles, can also predict the progression to a disease. So that is why it's basically a dynamic process, and that reflects an organism's response to various conditions or its environment, and hence it's useful for studying the pathogenesis or drug resistance mechanisms. And basically, transcriptome is from the RNA, because transcript is RNA. Transcription from DNA to RNA is the process of transcription. In order to study the transcriptome, you need to begin with the RNA, and it's very important to have a good quality RNA, and that RNA profile you can visualize either by gel electrophoresis, where you would expect two clear bands of 18 and 28 as rRNA, or in bacteria you would see the bands of 16 and 23 as rRNA, and that would indicate that the RNA is of good quality, and that's generally visualized by some instruments also. It's very important to have a good quality RNA for a good quality transcriptomics experiment, because otherwise if it is degraded, you cannot compare it between two different conditions. So one very common approach for transcriptome analysis is microarray. You can study some small number of transcripts by reverse transcriptase PCR also, but for entire transcriptome or for large number of transcripts, you would need microarray. And in microarray, the mRNA is converted into labeled cDNA, and competitive hybridization on a chip is done, followed by scanning to see which type of label is showing higher intensity of signal. Whereas the RNA sequencing is basically sequencing of RNA or the mRNA. But mRNA is first converted into cDNA and it is sequenced. How it is sequenced? So once mRNA is converted into cDNA, it is like DNA sequencing. Once it is a double-stranded DNA, the adapters can be ligated to it, and this can be sequenced using next generation methods such as massively parallel sequencing approaches. So just a brief overview of the microarray experiment is that you first extract the RNA and then you convert it into mRNA. You first extract the RNA. It would contain rRNA also, and that need to be either removed because sometimes it is basically taking up more than 80% of the reads. In case of microarray, that is not a problem because mRNA is converted into cDNA using certain oligo-DT beads, and during that time it is labeled with certain dyes. So Psi3 and Psi5 dye can be used for labeling the mRNA or the cDNA from two different conditions, and it is hybridized on a chip where the higher expression would be quantified through higher signal intensity because its label would be more. And in the scanner, you basically quantify it. But for that, you need an oligo chip on which the oligos of predefined sequences are spotted. If an organism is a new species for which you don't know the genome, then this approach would not work. So that is one of the major limitations. Besides, the dynamic range of microarray is very low. What is the meaning of dynamic range? For example, so here you basically quantify based on the color intensity. So the color scale can be of some limited resolution. Whereas in RNA sequencing, that we come in the next slide, you can quantify it more precisely. So the dynamic range is a major limitation with the microarray for the genes which are either very highly expressed or for the genes which are expressed at a very low level. So for such conditions, RNA sequencing has an additional advantage. Besides, you don't need to know the genome sequence prior to an RNA sequencing experiment, which is not the case with the microarray. For microarray experiment, the basic requirement is that you need to have a chip on which the oligos are already spotted, to which the cDNA molecule will hybridize. But in case you have a novel species, you cannot do that approach. So, okay. So Prashanth, just maybe 2 or 3 more minutes, and I would finish and will hand over to you. So just summing up the NGS for transcriptome analysis, that is actually referred as RNA sequencing. It's actually not RNA sequencing, but cDNA sequencing. So in that you basically have 2 different conditions. For example, one normal tissue, another is a tumor tissue. You isolate that entire RNA and it's converted into cDNA. The cDNA is added with some adapters, and those adapters allow the sequencing. And based on the sequencing, you identify what is the sequence of that particular gene, particular transcript. And that allows you to identify that that was the expression of this particular gene. More the number of the transcripts, there would be more number of the reads. And that is how it is quantitative. So this enables detection of transcript isoforms, which were previously not known. Whereas in microarray, you have to spot all possible splicing variants in order to identify those. But in RNA sequencing, you don't have to have the prior hypothesis or information. So that is one important advantage of RNA sequencing. And as I mentioned about microarray, which has lower dynamic range, in this RNA sequencing, you have a broader dynamic range. And it can quantify low expressed or the highly expressed genes very precisely. It also allows higher level of multiplexing because you can have indexes on the library molecule. So, for example, this type of library molecule, as Prashanth was also referring, you have adapters, and in the adapters you can have sequences which are from one end the P5 adapter and another end is P7 adapter. On both the ends you can have 6 to 8 bases which are defined or which are known to you. So for the sample 1, you have index number 1 for which the sequence of the index is known, ATGC, for example. And on the other hand, also you can have an index, and that allows you very high level of multiplexing, because with 12 indexes on one end and 8 indexes on the other end, you can have a combination of 12 times 8, that's 96 adapters, 96 dual indexes can be formed, and that can allow you to multiplex 96 samples. So you can, that is why get very high level of multiplexing. In addition, you can also investigate pathogen and the host genome interaction simultaneously, because the entire RNA contains, in case of a diseased tissue, it can contain the RNA of pathogen as well as host, and you can simultaneously identify what is the host pathogen interaction. So that is all from my side regarding the transcriptome, and there would be a talk on next week by Dr. Prakasha Kampaya from Mayo Clinic, Florida, on the application of RNA sequencing in malaria control and malaria pathogenesis. And there would be another talk by Dr. Senthil Rajagopal on CRISPR technology.